Okay. So what is topological data analysis? This is where the methods that I use come from. So unsurprisingly, this is rooted in the mathematical field of topology, which studies shapes. And it in particular considers things that are invariant in shapes under deformation. So you're allowed to stretch and bend the shape, but you're not allowed to cut or glue. And the kind of things that stay invariant are, for example, the number of connected components. So if your shape has two separate connected components, then no amount of stretching or bending will bring them to join together into one connected component or to fall apart into more connected components. Similarly, things that stay invariant are loops or voids in higher dimensions. And this leads to the often cited joke that mathematicians can't tell their coffee cups apart from their donuts. And this is because, as you can see in this nice animation from Wikipedia, you can continuously deform one object into the other. And the things that stay the same is you have one connected component, and then you have the handle of the coffee cup or the donut, which gives you a loop. And this also points to something that is functionally important for these two objects. Now, topological data analysis is the same thing, but for data. So now you're dealing with algorithmic methods to look at understanding shapes and surfaces in data. And for example, if you sample points from these two types of cheeses, then you will connect these points in a way that allows you to capture the shape of these cheeses, specifically that one has holes in it and the other one doesn't. Now, the method that is most often used in topological data analysis is persistent homology. So again, you're interested here in connected as loops and holes in various dimensions in your data. But in addition to just saying that they're there, you also get a notion of how persistent they are across multiple scales, for example. You're looking at local information in your data set to get an idea about the global properties. And what is really nice in the context of networks is that you can include more than just pairwise connections. So what does it mean for a data set to have shape? This is something that as humans, we quite intuitively see. So if I give you this data set and ask you what shape it has, you will probably tell me it's a circle. And this is exactly what we're looking for with persistent homology. And the way we do this is that we have data points and we blow up little epsilon balls around each data point. And as soon as two of these balls intersect, we add in an edge. If we have three pairwise intersections, we add in a triangle. If we have four, we add in a tetrahedron, and so on. So what we're building here is called a simplicial complex, and you can think of this as being a scaffold of shape built on your data points. So right now we can't see the circle yet. So we need to choose a larger epsilon, and then we start seeing this circle. But if epsilon becomes too big, everything is covered in higher order structures. So what does this mean? How do we choose our epsilon? And the key idea behind persistent homology is that you don't choose one value of epsilon, but you choose an increasing sequence of these radii. And then what you have is a filtration of a simplicial complex. And you can study the changes in the topology along this filtration. So you're going to look at connecting components, loops, and voids as you build this filtration from your data. Now, there are lots of different ways to create such a filtration. I showed you one example, which is probably the most common one, which is the Vittorius-Rips complex. But the tools are applicable to any filtration that you come up with from your data. So the Vittorius-Rips complex, more formally, you choose a sequence of increasing distances epsilon I. And then in the ith filtration step, you define your k simplices to be the unordered k+1 tuples of pairwise distance at most epsilon I. The output from this procedure is very nice. It's called a barcode, and it's a visual representation of what happens to the topological features in the filtration. So in dimension 0 here, we're looking at connected components. And on the x-axis, we have the epsilon value. And so here, each bar represents one connected component. So initially, each point is a separate connected component. And then as we add in more edges, we have bars that end because the corresponding connected component has merged with another connected component, and so it kind of lives on in the bar that is associated to that other connected component. So here you can see at around epsilon 0.6, we just have one connected component left, and this then continues on until the end of the filtration. In dimension 1, what we're monitoring are loops, so exactly this kind of circle that we set out to look for. And the bar starts when we first see this loop in the filtration, and then it ends when it's covered by triangles, tetrahedra, or the higher order simplices. And so the length of this bar tells us how persistent this loop is across the filtration. So how many scales we can traverse with our epsilon values and still have this loop in the data. And kind of the common philosophy behind persistent homology is the more persistent a particular feature is, the more important it is for the overall shape of the data set. But there are examples when this is not necessarily the case. Is so far everything clear? Okay. Right, so you might think, okay, this was a very artificial example. When do we actually get data points on a circle or even on more complicated shapes? And it turns out that real-world data has a lot of shape. So we have one really nice example from 2022, where Gardner et al. Looked at the activity of grid cells, and it turns out that this lies on a torus, and they were able to find this using persistent homology. Then another example that is very popular among people working in topological data analysis is the conformation space of cyclooctane. So this is a molecule that consists of 8 carbon atoms, which are connected in a ring. And depending on the chemical and physical forces around these molecules, it can take very different conformations in space. So it can look kind of like this bathtub shape, or it can have more a chair shape. And you can map out all of these different conformations into a 24-dimensional space by taking the 3D coordinates of the carbon atoms and then just writing those down in a 24-dimensional vector. And so each of these conformations now corresponds to one point in R24. And it turns out that these points lie on a sphere intersecting with a Klein bottle in 2 rings, which is quite amazing. And here you have a projection down into 3D or 2D here on the screen of this.
What does that tell you?
So I've spoken to chemists about this. And it turns out you have sort of certain symmetries which lie either on the sphere parts or on the Klein bottle parts. And sort of in these intersection regions, you go more from one type of symmetry to another.
Would also tell what the role of being the torus in the grid cells, like the function of grid cells and why it would be in the torus?
So it's kind of to do with the periodicity of when the grid cells respond. So you have periodicity on either end, kind of a domain on the top and the bottom and the left and the right, and this is reflected in the torus.
And what's the function of grid cells?
So they're, as far as I know, for space orientation.
But it makes sense today.
Yes, yeah. It's something that totally makes sense within neuroscience. But that was actually difficult to look at using conventional methods. Yeah.
So the idea here is that we try to find some like topological properties, but that in principle tells you absolutely nothing unless you ascribe meaning to those properties.
Yes.
You have to find something else that then gives meaning to that.
And that is a very important part. Yes. So especially in biology, you need to know why shape would be relevant in the context. I mean, you can throw any of these methods at any data set, but then the question is, what does it actually tell you? And this is something that you can kind of encode in the filtration, what properties you want to look at, and then it becomes interpretable. Yeah.
So for the grid cell, I mean that every grid cell receives some orientation, and then because this is continuous, you go in a torus or something like
yes, that. As far as I understand.
Okay.
Yeah.
Okay. Nice.
And then the final example, which we'll also look at further in this talk, are tumor blood vessels. And here, the shape of these vessels changes as a tumor grows, also in response to treatment. And this is something that we can now quantify using persistent homology. So the general pipeline for persistent homology is we have some type of data. We have, for example, tumor blood vessels, but we can also have the output from experimental time series from neuroscience, which we can then use to build a functional network, or we can use the output from mathematical models. And then the key is really to find a filtration which captures the properties that we're interested in this data. And then there's a whole host of methods to post-process these barcodes. For example, you can use persistence landscapes or persistence images to vectorize them. And then you can use all sorts of statistical and machine learning tools on this output.
But if you're engineering the filtration to get the properties, I'm not sure what you gain with that, except to just say, hey, this property can be understood with topology. I guess the other way around is more interesting if you have a data that you have no clue what it is. And then you try to find these topological things and then say, hey, there might be something here that is interpretable. Is that the goal rather than the other way around, engineering the filtration so that you recover a property that you already know? But
so with this, so topological data analysis can allow you to see things that are perceivable by the human eye, but incredibly difficult to quantify using conventional methods. So for example, in a tumor vascular data, I mean, I'm going to talk about this in a second, but the kind of quantifications that people used are length of blood vessels or thickness of blood vessels, and those don't capture anything about the connectivity or anything about the spatial arrangement. And this is something that we can capture using topological data analysis that just wasn't there before. But of course, the other way is also really interesting. If you find topological features and then you want to investigate what they actually mean in your data set. But it becomes very difficult because then you're kind of in the area what machine learning is doing a lot. They find differences, but they don't necessarily know what the biological underpinnings of these differences are. So it's really, in my case, I work closely with domain experts to really tease out what they want to know about the data set. And I try to think about how I can encode this in a filtration, which will then tell me something about it.
Maybe complement what Jay said, in topological data analysis, you have point clouds and distance. So that's the mathematical ingredients, but the distance are domain specific. So you need someone else to guess the best way to measure distance between points.
No, yeah, I understand. I was just going back to this case of the chemistry, because at the end you say, I found these structures, but then actually I could interpret this already because it's just classifying the different symmetries of those conformations. And then you don't gain much because you already had a quite good classification of those two different regions in terms of symmetry.
But you might, for example, be interested in when am I getting close to an intersection, and are there any sort of transitions that I can get by changing the forces minimally, where I get from one type of symmetry into another. So I think understanding this space is of value by itself. Okay, so why am I interested in tumor blood vessels? So as a tumor grows, initially it takes up nutrients from its surrounding by diffusion. And then at some point, this is no longer enough to sustain tumor growth. And this is when a tumor starts signaling to the surrounding tissue, to the healthy tissue, to start growing blood vessels into the tumor in a process called tumor-induced angiogenesis. This is the growth of new blood vessels from existing ones. So the tumor doesn't grow its own blood vessels, it really makes use of surrounding healthy vessels. It's one of the hallmarks of cancer, which means that it's one of the steps that is necessary for the disease to become malignant. And in particular, it's necessary for invasion of the tumor into other tissue or growth beyond a certain size. Now, functionally, these vessels supply the tumor with nutrients, but they also provide physical support for the tumor. They're involved in immune suppression around the tumor, and there's many more roles that these vessels have. Now, the process through which they grow is very messy. So there's really quick changes in the equilibria of pro and anti-angiogenic factors, which lead to structural abnormality of the resulting vessels. So in healthy tissue, you often find sort of mesh structures and hierarchical structures. But in vascular networks of tumors, you get much more messy structures where you have large invariant diameters. So you have this huge vessel down here and then tiny diameters over here. You have irregular branching. You can even have shunts between arteries and veins and loops forming. So from a perspective of blood flow, that's not a very efficient thing to have. And you can have heterogeneity in blood flow where you look at the same vessel at different time points and the blood is flowing in different directions. What is also very striking is that these vessels are highly tortuous. So you can see this here in these images of tumor vascular networks indicated by the yellow arrows. And this is also something that you don't find in healthy vasculature. Now, there's lots of questions surrounding these blood vessels coming from the biological side. So can we look at the vascular system of a tumor and use this somehow to predict its response to treatment, both chemotherapy and radiotherapy? Are there connections between the structure and the function of tumor blood vessels? Can we characterize heterogeneity in these networks and what treatments do to this? Do treatments reach all parts of the vasculature equally, or are some parts only going to be reached and others not? And is there a universal measure for all data sets, regardless of imaging technique, because each lab or hospital will have a different imaging technique? And can a process of renormalization, so when you start seeing less of these abnormalities over time, can this be quantified? And are there parameters that are dynamic and robust over time? And this is what I mentioned earlier. So what is done to answer all these questions at the moment in many labs is that they extract these networks from imaging data, and then they create histograms of lengths and diameters to try and understand what is happening either during tumor growth or during treatment. Now, from a mathematical side, mathematicians in the UK have been interested in this process since the 70s, and there's a huge zoo of mathematical models of tumor-induced angiogenesis. And just to give you an idea of how many there are, there's a review from 2017 that lists 12 different review articles on models of tumor-induced angiogenesis. And all of these models are wrong, but some might be useful. So how do you navigate this zoo? And actually, there's lots of questions surrounding these models that are of interest to mathematicians. So do the different models actually provide different results? Or in the end, are they all providing kind of a similar output? How sensitive are the results to changes in parameters? And then there are numerical challenges. So the simulations can be very expensive. So do we need to model this across a big domain, or can we just take a small domain and extrapolate from this? And is there a way to benchmark that uses information on network shape? And what is done in the literature so far is actually very similar to the biological literature, where people are looking at measures such as average cell density, vessel lengths, or diameters. You can also use indirect measures, so you can model oxygen distribution on top of these networks and then compare these. And it's actually only recently in this review article by Villanova et al., where they looked at graph descriptors and found that structural information such as the number of loops seems relevant to distinguish between different models and also within one model. So we wanted to address this question from a topological data analysis perspective, and the challenge here was really to find a filtration that would be meaningful for both the biologists and the mathematicians. And so the goals that we had was to quantify the shape of the vessels or of the tumor, to have a notion of degree of abnormality. And here we define this to be the tortuosity of the vessels and loops that are present in these vascular networks, and also the heterogeneity of these features across space. And then we also were interested in the degree of vascularization, which you can think of as measuring kind of the voids in this data.
And there was no way of quantifying that before? Is that
No. So not in the histograms that have been used before. And we compared actually our approach also to the standard approaches and found that it does distinguish better. Yeah. So there's some examples of similar applications from the early applications of topological data analysis, which I'll quickly go into now, which were an inspiration for what we ended up doing. And the first one is an example by Bendig et al., who looked at brain artery trees. And they were interested in sort of quantifying the bendiness of these arteries because this changes with age. And so what they introduced was they took as nodes and edges points that they sampled from these brain artery trees, and then the connections corresponded to actual connections of the points along blood vessels. And then they introduced sweeping plane filtration. So this means that they kind of went from right to left with a plane stepwise, and then introduced the edges and nodes that were on the right side of the plane to be in the filtration at a set filtration step, and then slowly slid this plane along and built those brain artery trees. And it turned out that in dimension 0, so in the connected components, they were able to actually measure the bendiness of these brain arteries, and this was correlated with the age of the subjects. Then Belki et al. Did a very similar thing with airways. So they used the sweeping plane filtration on airways, kind of starting at the nose and then going stepwise down. And they found that then the dimension 0 barcodes measured how bushy the airways were, how much they were kind of branching upwards. And they found that this filtration can correctly classify patients with compressive obstructive pulmonary disease and can distinguish between inspiratory and expiratory scans. And finally, the group that I'm in Lausanne at the moment, they were interested in quantifying the shape of neurons. And so they looked at a filtration by radial distance to the root of a neuronal tree, and found that this quantifies and characterizes structural differences between morphologically distinct groups of neurons. Now, what these examples have in common is that you have clear orientation, and we don't have this in the case of tumor blood vessels because they grow into the tumor from all sides. So you don't have any root or any tree-like structures. And so we weren't able to use any of these approaches directly. So what we ended up doing is we introduced the radial filtration. So we defined a tumor center, which we took to be the center of mass of points sampled from the blood vessels. And then we kind of blew up a sphere from the center outwards, increasing this radial distance stepwise, and then included all points and edges that were kind of within the sphere at a particular step in our filtration. What does this measure? In dimension 0, it should give us a notion of tortuosity, similar to what we saw in the brain arteries. And in dimension 1, because we have loops in this network, we can measure these loops, and we also get an idea of where they are in relation to the tumor center. Why do we measure tortuosity? So we introduced a new measure by looking at how many short bars we have in the dimension 0 barcode. So these correspond to parts of the vessels that you kind of cut by this growing sphere, which correspond to short connected components, and then quickly merge with other connected components around them. So they have a short persistence in the barcode. And we take this normalized by the number of vessel segments to take out the effect of the size of the networks. To compare to standard ways of measuring tortuosity, we also looked at the chord length ratio, where you look at the distance between 2 points and then look at the path distance between them as well and take the ratio between these. And you can also measure, for example, the sum of consecutive tangents of their angles to get an idea about tortuosity. These are measures that are used in practice and give sort of mixed results and capture slightly different aspects of the tortuosity. And so we kind of introduced a new one using topological data analysis. Now, I also mentioned that we want to capture voids, and so far we only have edges and nodes coming from the vascular network. So where are the voids coming from? So for this, we use a slightly different construction, which is called the alpha complex. We took points that were sampled from the blood vessels, and then you build a Delaunay triangulation on those, and you introduce the simplices roughly by distance or by size. And this gives you a notion of voids or the size of voids in dimension 2, which is captured by the barcodes. So the pipeline was we had the vascular network data. We used the radial filtration and the alpha complex filtration to create barcodes. And from these barcodes, we extracted topological descriptors of the data, namely in dimension 0, we had a tortuosity descriptor. In dimension 1, we looked at the number of loops and the distribution of these loops across the networks. And then in dimension 2, we looked at the persistence of these bars to get a notion of the size of voids. We had two different types of data coming from amazing experimental collaboration. So the first one was a collaboration within Oxford, where they were able to look at the same tumor in a mouse over several days. So this was intravital microscopy, where you can see really the vascular network growing in a tumor. And so this was live 3D imaging through a window chamber implanted on the mouse's back. And this gave rise to longitudinal data. In the second case, we had ultramicroscopy data from a collaboration with the pharmaceutical company Roche. Here, for every tumor, we just had one snapshot in time. But here they used optical sectioning with a laser, so we were really able to reconstruct the full 3D networks of these vascular, the full 3D vascular networks of these tumors. These tumors were subjected to different treatment conditions, so there were controls. And then in the Oxford data, we had vascular targeting agents, both to introduce more abnormalities and to reduce abnormalities in these vascular networks over time. This was kind of to see if we could capture what you would expect in this data. And in addition, they also had treatment with radiotherapy, both single dose irradiation and fractional dose irradiation, where it's not as well known what this does to the vascular networks.
Just to make the parallel with the math, so what is your expectation given your definition? So when this H0, H1, H2 grows, the tumor is worse or better. So what's your expectation? So
for the tortuosity and the loops, because these are measures of abnormality, I would expect with the vascular targeting agents that increase abnormality to see an increase in tortuosity and an increase in the number of loops. And in the case where we have more normal looking networks, I would expect those to go down. Yeah.
And for the voids?
For the voids, you would expect larger voids if you have more avascular regions. So if kind of the network vanishes and parts a little bit more. So that's also when you would have treatment that attacks the network directly. Yeah. And then in the Roche data, we just had, again, an agent that slowed vessel growth, leading to more normal networks versus controls. And just to show you, so these are wildly different data sets from two different imaging modalities. So on the left, you have this intravital data set, which is dynamic from Oxford. And on the right, you have the ultramicroscopy data set. So even like the size of these networks is wildly different. And also to show you that you can see these treatment effects by eye. So on the top row, we have a control on day 3 and control on day 7 in the Roche data set. And on the bottom row, we have a treated tumor on day 3 and day 7. And you can visually see that there are really changes in the networks that we should be able to capture. Okay, so let's see some results. So this is from the Oxford dataset using the data on vascular targeting agents, where we know what to expect in terms of the trends. So we can see here the days since treatment, and it's all normalized by day 0 when the imaging first started and when treatment was given. And we have the controls in beige, and then in pink we have DC101, which makes the vessel networks look more normal, and then DLL4 in pink in light pink, which makes the vessel networks look more abnormal. And you can kind of see on day 2 that there's a difference, especially between the more abnormal looking ones in light pink and the more normal looking ones in dark pink.
And just for curiosity, how did you set up the control, like to get the same area without tumor and target follow that area? Or
Sorry?
how did you set up the control?
So the controls were just not treated. So they were just observed. So you have the same imaging over the same time, but you don't have any treatment.
But still tumor.
Yes. So there's still a tumor. So it's still tumor vasculature, but it's kind of untreated, so it can do like a natural progression.
Yes.
On the level of loops, we see these differences much more strongly, especially then on day 3 and day 4. So we first have a change in tortuosity, and then we have a change in the number of loops on day 3 and day 4, which is very prominent between the 3 groups. Now, what we get in addition is that we get a spatial location of loops. So we can spatially resolve these treatment effects over time. So here we are looking at spherical shells around the tumor center, and then we're looking at how the number of loops changes in the specific spherical shells over time. And so blue is kind of stable or only slight increase, whereas red is quite a big increase. And you can see, so in the controls, we have kind of stable conditions on the outside initially, but then over time, you get more and more loops in all the layers. In DC 101, which makes the vascular network look more normal, the loops are kind very throughout the different layers. And in anti-DLL4, which makes the vascular network more abnormal, you have this very big increase in the number of loops in the outer layers of the tumor. Now, in the Roche data set, we looked at the loops and voids. And so here, we're not able to normalize by day 0 because we only ever see each tumor once. And again, in the controls, we have the loops are kind of stable over time, increasing slightly. In the treated cases, we have a decrease in the number of loops and an increase then on day 14, which is a much bigger timescale than we had earlier. And in the voids, you have, in the treated cases, these increase because you have less vasculature across the whole tumor, whereas in the controls, this is stable. And then to look at radiotherapy and how this impacts the vascular networks. So here we have the controls again in beige, and then fractionated irradiation. So this is small doses over 5 days in brown, and then in purple we have single dose irradiation. And you can see that there's really a difference also between these two treatments in the level of tortuosity. And then on the level of loops, you just have huge variability in the single dose irradiation, whereas the fractionated dose radiation seems to be more stable. So that was very interesting for us to see, especially because in the literature, there's a huge, so there's very conflicting results whether radiation therapy makes vascular networks look more normal or more abnormal over time. And again, we're able to spatially resolve this. And so in the fractionated irradiation, we have stabilization sort of on the core of the tumor, but then on the outside, we actually get an increase in the number of loops, whereas single dose irradiation, you have an increase starting on day 3, sort of almost throughout the tumor, apart from right in the center. And just to show you a comparison to standard measures, so something that is standard to measure is like number of segments or number of branching points, or the volume, vital volume, necrotic volume of the tumor. And all of these measures are highly correlated. So red means that you have a high correlation, whereas the topological measures here form their own small group of measures that capture complementary things to the standard measures. Okay, are there any questions so far? Okay, so we saw that topological descriptors support the proposed action mechanisms of vascular targeting agents and can capture effects of radiation therapy. We can spatially resolve these, and the alpha complex can quantify voids, so avascular regions. And so now we thought, how about those mathematical models? Can we use similar approaches here? And we chose for this the Anderson-Chaplin model because it's very widely adopted in the mathematical biology literature, and it's one of the simpler models that at the same time produces very interesting output. So the general setup here was, in this model, you have two biological effects that are taken into account. One is chemotaxis, so you have you have a vessel on the left of the domain, and then you have a tumor on the right, which sends out chemoattractants and makes those vessels grow towards itself. So this is done by chemotaxis. And then you have haptotaxis, which kind of distracts the vessels to go more towards each other, or towards fibronectin, which is bound across the domain in the extracellular matrix. And depending on how you tune these parameters in the model, you can get visually very differently looking output. And so the idea here was, okay, we can use topological data analysis in experimental data, but can we use it to distinguish between different groups of output in synthetic data? And we also compared this again to standard descriptors, and then saw if we could cluster similarly looking simulations outputs
I
together.
have a question, Nixon, before you say that and what you showed, like,
Mhm.
because in a way, as Jay saw in the beginning, like you create some topological features that would be nice to detect the vascular networks and change in the tumor networks compared to control and treatment. But could also you go back and tell the mechanisms only from the topology?
If you know what the topology is capturing, then yes, because like we were able to say it's the loops in this case, or it's the voids, it's the connected components. If you did say a Vittorius Rips filtration, it would be much harder to say what this actually tells you.
So could you have formulated your question now just with topology? Now that I have this radio filtration, the tumor is bad and this goes bad, but can you formulate the mechanisms from the topology?
Not necessarily, not by itself. So in connection with mathematical modeling, I think that would be a step towards really understanding the mechanisms. But you can say structurally what is happening. And you can, for example, so we had a different project where we looked at topological descriptors and geometric descriptors, and we found that those could be predictive of how well a tumor responds to radiotherapy. Because a lot of the things depend on how your network is initially when you apply the radiotherapy. So there's a huge heterogeneity in response. And we looked at how topological descriptors could capture this heterogeneity. And it turns out that some things are really helpful in predicting how blood flow will react if you start applying radiotherapy. Yeah. But this was synthetic, so this was in mathematical models. So we're still quite far away from actually doing this in clinic or in experimental data. Okay, so here you can see example outputs from this model. And again, visually, they're very different. And if you now apply standard biological descriptors, for example, looking at the number of tip cells here, or looking at the length of the longest vessel, then you can try and cluster the outputs coming from different combinations of the chemotaxis and haptotaxis parameters. And you don't really get much of a stratification here, maybe a little bit in the case of the length of the longest blood vessel. If you apply topological descriptors, then the parameter space gets stratified very nicely into different groups. And we looked at representatives of each of these groups, and they actually correspond to differently looking outputs from the simulations. So again, the TDA is able to pick up something that the standard descriptors are not. So we found that the topological descriptors reveal distinct parameter regimes in this model, and visually similar output is clustered together. And we think that this paves the way to investigate connections between structure and function more, because we can now quantify the output from the models as well as the experimental data. Now, something that I've been thinking about more lately is data coming from spatial biology. And so we've seen unprecedented resolution in new experimental techniques coming from biology. And one of the goals from these experimental techniques is that you can finally precisely characterize the tumor microenvironment, so the tumor and sort of the immune system around it, the vascular system around it, the cellular interactions in there, and the local architecture. However, we're quite far away from actually being able to do that because the methods that are currently used don't really fully exploit the available spatial information. So typical statistics that are being used are cell count, so similar to the histograms that we had earlier in the vessel networks, distances to nearby cells, or then machine learning, but there we don't really get a lot of interpretability, although this is changing a little bit now. So we thought topology might be able to capture something that these methods don't while bringing interpretable output to the table. Mathematically, what is interesting here is, so typically in persistent homology, we think about data points that are all of the same type. So we have a single system of data points. But if we think about these spatial methods coming from biology, this corresponds to a multi-system of data points. So you have data points that they also have different types. And so how do we deal with this in a topological data analysis framework in a way that can actually capture spatial relationships?
Different types.
So if you think about spatial biology, for example, they have imaging techniques which reveal not just that you have cells, but also what type of cells. So here, for example, you could have a tumor, and you could have in yellow the blood vessels around it, and then immune cells which travel between the blood vessels and the tumor. And so you end up with these different types of points.
Basically, that's just a higher dimensional data set, because we just add another layer.
Yes.
Color.
Yes.
Is that what it is?
It is, but so if you then treat this with conventional topological data analysis tools, you just get an idea of the overall shape of this, but you want spatial relationships to be encoded, and you want to know which cells, which of the cells.
So you mean in the standard persistence, you get distance between points, but now each type of point has a different color, so you need multi-parameter to get distance from points with different colors or something.
Yes, so you don't need multi-parameter. We've avoided multi-parameter, but you need a simplicial complex that captures those interactions. Yes.
Do you want like linking numbers then, or?
What do you mean by linking numbers?
Well, I mean, if you have like a loop between the red points, so that kind of links with the yellow points.
Mm-hmm. So that is something that we're capturing. So kind of shared topological features. So a loop that is shared between the red points and the yellow points, for example. Yes. So it's that kind of information that we're looking for. So here we again use the output from a mathematical model to fine-tune and test the methods. And this was an agent-based model where effectively you have a tumor and you have blood vessels around it which produce immune cells, and then these immune cells can interact with the tumor. And depending on parameters that you fine-tune for the immune cells, you can get three distinct qualitative behaviors. So you can have elimination of the tumor, where the immune cells eat up the whole tumor. You can have equilibrium of the tumor and the immune cells over time, and you can have escape of the tumor towards the blood vessels, which is a precursor to metastasis. So at the very least, we wanted our methods to be able to distinguish between these three qualitative behaviors given an image coming from this agent-based model. And the other thing that the model captures is there are different subtypes of immune cells which change over time and are also characterized by where they are spatially. And can we determine if a particular image comes from a simulation that is dominant with one type of immune cell versus the other without knowing which immune cells we're dealing with? So these were the two questions we wanted to investigate, and we created two different topological tools. The first is based on the Dowker complex. This looks at pairwise relations and shared features in terms of the topology between them. And here we used a vectorization that kind of emphasizes these shared features to look at the dominance of one immune cell type versus another. And in the second case, we created something that was based on the witness complex, where you take into account multiple relations at once, and you create distance vectors to look at really the distances of the spatial distributions of these cells, and then use this for clustering and see if we can recover the three qualitative behaviors. So in the first method, which is Dowker persistent homology, this is based on the Dowker complex. So if you have two different types of points, say U and V represented by these dots and crosses, then you can choose one of the points as being your potential vertices for a simplicial complex. And then, as before, you start blowing up epsilon balls around each data point, but now you only connect intersecting the data points from intersecting epsilon balls if in the intersection you have a point of the other type as well. And this, it turns out it doesn't matter which points you choose as your vertices, because in the end, the persistence barcodes are going to be the same. So we did this in the output of the model, and we also compared this to standard topological features done separately on the different cell types. So we can see these here in green, and then in gray we have these new features, and a lot of them outperform the standard features which don't take spatial relations into account, some don't. And if you use standard features, so distances to nearby cells, you only achieve about 80% accuracy when looking at which immune cell type is dominant in a particular image. So we really seem to be capturing something that is relevant to this task using this construction. In the second case, which is based on the witness filtration, so we have this point cloud with multiple types, and we choose one type of points to be our reference points. We build a Delaunay triangulation on this, and then we use the other points as witnesses for different simplices. So we look at how many, so we look at the closest point of the reference cell type from this yellow cell type, for example, and we assign this as being a witness for this particular point and also for the simplices arising from this point. And then we conversely look how many witnesses do I have for a particular edge node and triangle, and we bring them in by the number of witnesses. So for each witness cell type, we then get a different filtration of the same simplicial complex. And the idea is that then you can take distances between the barcodes, which really reflect the effects of the different witness cell types. So if we then use these distance vectors for clustering, on the left, we have expert annotation from this model. Then on the middle, we have fully automated clustering on the distance vectors from the multiplex witness filtration. And you can see that there's quite a good overlap in this. The only differences are kind of in these boundary regions between different qualitative behaviors. And if we look at consistency across 20 simulations from each parameter pair, then you get quite high consistency sort of within these qualitative behaviors. But then on the boundary regions, you get less consistency, which is what you would expect. And actually, if we look at 4 clusters, then these boundary regions form their own cluster using these features. So we thought that this was very interesting. And we're able to really capture spatial relationships going beyond single system methods using these new tools. And the Dowker features predict dominant macrophage subtypes, while the witness features capture qualitative behaviors of the model. Okay, so in the final few minutes, I'll just quickly go into local persistent homology. So one of the things that is challenging in persistent homology is that it doesn't scale well to large and particularly noisy data sets. So it's very difficult to compute in many cases. And in such cases, it can be interesting to pre-process the data and, for example, select landmarks that are relevant in this data set to perform the computations on. And so far, there wasn't really a good way of doing this. So there was random sampling and maximum sampling, which kind of gives you a good sort of maximally stretched out landmarks across the data set, but it also emphasizes outliers here. So something that I did, where I'm not going to go into too much detail of the math, is use the Maya-Vittori sequence as a motivation to look at local computations of persistent homology, where we don't have issues from the computations, and then look at the barcodes, which can be very different for different types of points, and take out a measure to tell me how much of an outlier a particular point is. So here we have data sampled from a sphere. The sphere would be considered to be the signal, and then noise around the sphere. And if you have a sphere point, then you typically have barcodes with sort of short bars because there's many points on the sphere. If you have a noise point, which is further out, you tend to have longer bars. And if you have points that are very far out, you don't have any bars. So we don't consider these to be particularly relevant. And then we look at the length of the longest non-infinite bar in dimension 0, 1, and 2. And we look at these scores that we get from there. And it turns out that if you select points where locally you have low scores, then you get particularly many sphere points in your sample. And it turns out that this is also the case for low sampling density. So here we're looking at the fraction of sphere points versus the sampling density. And in black and in red, we have persistent homology-based scores with which we select data points as landmarks, whereas the other lines are.
How do you define the scores?
So I take the length of the longest non-infinite bar in dimension 0, 1, and 2. Yeah. And this, so I don't have time to go into this too much, but this kind of is inspired by the Maya-Vittori sequence, and it tells you something about how relevant a particular point is for the overall persistent homology of the data set. Yeah. And so it turns out that these really outperform the existing sampling techniques, just taking this local persistent homology into account. Now we can use a similar approach to work with data that doesn't satisfy the manifold hypothesis. So the manifold hypothesis is something that underlies very many data science techniques nowadays. And the manifold hypothesis states that data points typically cluster near some unknown manifold of dimension substantially lower than the ambient dimension of the data. And it's a cornerstone really of modern data science. It underlies manifold learning, dimensionality reduction techniques, PCA visual perception, subspace clustering, generative adversarial networks, and many more. Now, data can also be sampled from non-manifold or singular spaces, and so there you can't really use these methods. The simplest example is the union of two manifolds along a common submanifold. So an example would be the cyclooctane data set. And this can, for example, arise if you have data sets with more than just one class of data. There's very few techniques to analyze such data and find singularities. And so what we started out with is that manifolds of dimension n locally resemble an n-dimensional Euclidean disk. But of course, persistent homology doesn't necessarily, or homology in general, can't really distinguish disks, but it can distinguish n-dimensional spheres, because the dimension of the ith homology is 1 if I equals n or 0 otherwise. And the boundary of n-dimensional spheres are n-1. N-dimensional disks are n-1 dimensional spheres. And so what we did was we approximated kind of this boundary by looking at annular neighborhoods around each point and then computing the local persistent homology of these annular neighborhoods. The idea being that if you have such points sampled from intersecting planes, then if you're at an inner point in such a plane, you will have one bar in dimension 1 here coming from this annulus. If you're close to an intersection, you will have at least 2 bars. And if you're far away from an inner point, far away from an intersection, so close to the boundary, you will have no bars at all. And just by looking at the number of bars in the dimension 1 barcode gives us, sorry, it gives us like highlights different types of points in this data set. So points in red close to the intersection, then in green close to the boundary, and uncolored if we're on the inside of one of the planes.
Yeah. Sorry, how this would relate to if you begin of your talk when you make the centroid. So if you get the centroid points, you would lie already in the intersection. Would that also work nice for this kind of detection?
What do you mean by the centroids?
So in the tumor
Mhm.
part of your talk, you consider the centroid of all the vessels.
Mhm.
In the SD specific manifold, the means in the intersection, maybe by chance, maybe the intersections are different. So in that case, would also this type of filtration detect the intersection? How?
So if the centroid is close to the intersection, then in that type of filtration, you might detect it. If you think just about this example and start sort of building a sphere from the inside outwards, you would just have a connected component, right? So you wouldn't really have any dimension 1 signal. So you wouldn't necessarily have it, but there are certainly cases where you could have that. Yeah. But also if your intersection doesn't lie sort of in the centroid, so here with the cyclooctane example, we have these 2 rings, then this is also detected by local persistent homology. And we also looked at the Henneberg surface, which has a lot of self-intersections. And here again, we're able to find points that are close to the intersection. So not precisely the intersection, but close to, and also points close to the boundary. And it turns out, so the standard method that was used before to detect such points was local PCA. And so here again, you choose a local neighborhood to do your PCA in. And it turns out that local persistent homology is much more stable to the choice of radius. So here in orange, you have local PCA, and you have the Hausdorff distance to sort of reference points which are close to these singularities, and then you start increasing the local neighborhood in which you perform calculations. And you can see that this Hausdorff distance goes up very quickly, which is because it starts including points that are in regions that have high curvature, whereas local persistent homology is agnostic to such things, so it's stable for much longer. And when it does start including sort of nonsense points, it's not because of local curvature issues.
One question. I really like the example because you really show the intersection matters and you can detect the persistence. But what happens if I get data that is empirical and I don't know the underlying manifold, but maybe there is an intersection? Is there a marker that you say, oh, this type of pattern in the persistence indicates that the underlying manifold may be in an intersection, or is it still not possible to answer that?
Not at the moment. So there are now other methods that look at how Euclidean the neighborhood is, for example, or then a persistent homology independent method, which might be able to do that. I'm not very familiar with that. But like from these local computations alone, I don't think you'd be able to do that. It's also a bit dependent on the sampling. So I'm working on an example right now with collaborators where we're having a bit more issues in sampling nicely from that space. And so if you get very dense, very non-dense towards the boundaries, then it's much harder to detect. Okay, so local persistent homology can detect points close to geometric anomalies. It can be helpful for selecting landmarks, and it's more robust to the choice of neighborhood, radius, and surface curvature than local PCA is. And I'm sure that there are many more future applications that we're yet to see.
So geometric anomaly, you just mean self-intersections?
Intersections and boundaries, yes, just singularities, yeah. Okay, so this brings me to the end of my talk. So thank you very much for listening. And I would like to thank my many collaborators from Oxford Mathematics. So Jay, Iris, Helen, Heather, Vidit, and Jared. And then also from the biological side, from Oxford Radiation Oncology, Jakob, Bastian, Russ, and Ruth. And then from Roche, Florian, and Francisca. And on the Anderson Chaplin model output analysis, John Nardini and Kevin Flores, and lots of different funding bodies. So thank you very much for listening.
I have one question. I like the idea of the centroid distance, also because it was inspired by actual structural data. But do you think this is a good way to go for functional data? Because we could just project correlations in a space and then just make a filtration from the centroid of that space. Does it, because the normal filtration correlation data favors clustering of very close neighborhoods, whereas what you say, you have no bias in the cluster. The cluster really thinks that could be really far in your correlation space. So do you think it is a way to go to make this type of filtration non-Euclidean data?
That could be interesting. Yeah, I haven't done it myself. I'm only familiar for correlation data sort of with the clique filtration, but it could certainly be an avenue to explore. Yeah.
Just to make sure if that I understood correctly. So when you started the talk, you said that around each point you consider epsilon, and then as the epsilon grows, you connect two dots, right? But later on, you said that there could be also other measures, so you like when you do topological data analysis, you try to define. So not only like that small epsilon, but also different measures by which you can quantify the data.
Yes, so for example, in a sweeping plane filtration, so in a filtration, you just build your simplicial complex, and the way to build it is defined by what you want to capture. So if you want to capture distance-based things, then the Vittori-Rips complex with the epsilon balls is a good idea. If you want to capture more sort of curvature-related things, then the sweeping plane approach is interesting because you start building your simplicial complex sort of from one side to the other. And so you're no longer distance-based, but you're more kind of spatial location-based. So it really depends on what you want to emphasize.
So on the question that so like the biological information that I want to get out of the data.
Yes, yeah.
No more questions, then let's speak again.